Thaise Carneiro, as Patricia and Thaise. I'm also from Brazil, and I'm currently working at CHOP. I'm a postdoc fellow at the Center for Applied Research, Applied Genomics, CAG, at the Children's Hospital of Philadelphia. And I work under the supervision of Dr. Håkon Hakonarsson and Dr. Marina Bakai. And today, I'm going to present the mouse model for the CHAMP1 gene therapy that we are developing at CHOP. So just to give a brief overview about the CHAMP1 function, CHAMP1 is the short for chromosome alignment maintained phosphoprotein 1, which is located at chromosome 13. CHAMP1 has 3 coding regions called exons, and this is the model, the 3D model for the CHAMP1 protein that has 812 amino acids. CHAMP1 protein is essential for the division of the cells and also the segregation of the genetic material. So it guarantees that the right amount of the DNA go to each new cell. And both processes are important to the development of the brain. Also, as all the doctors are told us today, CHAMP1 also interact with 2 main proteins, which is the MAD2L2 or F7, and also POGZ. And both proteins also collaborate with CHAMP1 during the cell cycle and to guarantee the right division of the chromosomes. So here is the structure of the CHAMP1 protein, just to show you that here is all the mutations already reported until now in the patients. And CHAMP1 has zinc finger domains, 5 of them, which is represented by the black box here, which is important for the protein function. So CHAMP1 is expressed during fetal brain development and throughout all fetal and adult tissues. Recent research also showed that it's involved in cell survival through the expression of anti-apoptotic genes and proteins like the BCL2 family, and is yet to be discovered if the role of CHAMP1 in cell survival is related to the pathogenesis of intellectual disability. Here, briefly, just to tell that the main symptoms of the CHAMP1 mutation is developmental delay or intellectual disability, and also severe speech delay or loss. Also, the kids that have the alteration present a variety of symptoms like learning disabilities, feeding difficulties, anxiety, abnormal MRI, also visual abnormalities, motor delay, hearing loss, and others. So having this CHAMP1 background in mind, I'm going to present now about our project at CHOP, which is the characterization of the novel mouse model with 35 base pair deletion in CHAMP1 gene and exploration of the adenovirus gene therapy to correct syndromes arising from mutation in the CHAMP1. So in our model, we are using the C57 strain for the mice. And what happened with these mice is that it has a deletion on exon 2. So this deletion is made using a technology called CRISPR-Cas9. In this technology, we cut a target region of the CHAMP1 gene, in our case, a part of the exon 2, and then this deletion stays in the gene. So as I said, the technology is CRISPR-Cas9. So the CRISPR-Cas9 methodology was performed in mouse zygotes, made in this 35 base pair deletion on the exon 2. And we obtained our strain, our mouse model, from the Mutant Mouse Research and Research Center at UC Davis. We started our colony with 4 mice with the deletion at CHAMP1 gene in heterozygous. Our first strategy to breed our colony was to cross the males and the females, both with the CHAMP1 deletion. However, when we tried this way, the females present dystocia, which means difficulties in giving birth, and then we decided to change the strategy. In our new strategy, we are crossing the CHAMP1 mice with the deletion in heterozygous with wild-type mice. In this case, following the Punnett square, we have that the wild-type has 2 exon 2, normal for CHAMP1, And the CHAMP1 mice has one exon 2 with the 35 base pair deletion and one normal exon 2. According to the probabilities, when we cross those mice, it's expected that half of the prol to be heterozygous for CHAMP1 deletion. And when we changed the strategy, the mice started to breed better than the previous one. So our project is divided in two phases. On phase 1, we are going to characterize the CHAMP1 mice comparing against the wild type through behavior tests and molecular characterization. At phase 2, we're going to treat the CHAMP1 mice with the adenovirus and then repeat the behavior test and also the molecular characterization to assess if the vector was able to rescue the phenotype and also the molecular profile. So on phase 1, we are going to characterize these mice comparing these two groups, CHAMP1 against wild type. We're going to perform mice phenotype characterization through behavior tests. For example, the novel object recognition, which will assess the memory of the mice, IMAIS, which will give us information about anxiety and spatial learning. Also, the response of the mice to adverse cues in the environment, and measure the strength using, for example, the grip strength test. With all the behavior tests, we're going to have information about anxiety, strength, cognition, memory, and spatial learning ability for both groups. And then we are also doing on phase 1 the mice molecular characterization, where we're going to assess the CHAMP1 gene expression in both groups, also CHAMP1 protein expression in different tissues, like spleen, pancreas, ovaries, testes, hearts, thymus, brain, mainly frontal cortex and cerebellum, skeletal muscle, and stomach. And we are also going to check the interaction of CHAMP1 with other target proteins like MAD2L2 and POGZ for both groups. On phase 2, we are going to inject the CHAMP1 mice with our vector using the adenovirus. And in our case, we are using the AAV9. This adenovirus was already used for mice therapy before to treat heart disease with high success. And this adenovirus leads to a high level and stable gene expression, also has tropism for the central nervous system, and leads to a low immunoresponse. Our adenovirus is going to carry the normal copy of the CHAMP1 gene. So this adenovirus is going to inside the cell, mainly the nucleus, and then it's going to release the normal copy of CHAMP1 gene, leading to the normal expression of the CHAMP1 gene and protein. And we developed the vector together with the University of Pennsylvania, mainly the PENCOR. So just to update the mice project until now, we started around March this year, and we are breeding the colony. As I said before, our first strategy, the females had dystocia, so we had low breeding rate when we crossed CHAMP1 heterozygous, the male with the CHAMP1 heterozygous female. Then when we changed the strategy, we have a better breeding now. However, we needed more time to reach the number of CHAMP1 heterozygous to the study. Until now, we have 78 mice, 31 CHAMP1 with the deletion in heterozygous, and 45 wild types between 8 and 19 weeks. We also need 20 mice with 10 weeks at once to start the behavior test for both groups. We are in phase 1, and phase 2 is scheduled to start as soon as the behavior tests are done. Also, all the molecular characterization of the mice, the expression of the protein in different tissues was already done. So meanwhile, when we are breeding our colony, I started an experiment to test, to check the vector assessment using cell lines, to see if our vector was working to rescue the CHAMP1 expression. For this, I used the SH-SY5Y cell lines, which is a human neuroblastoma line established in 1970 from a metastatic bone tumor from a 4-year-old cancer patient. And this cell line was used in the literature for many research, for 3D cell culture, high-throughput screening, immunology, neuroscience, and toxicology. So other reason for us to choose this cell line is in our checking, we detect that the SEH-SY5Y was one of the cell lines that we have available in the lab that had the highest expression of the CHAMP1 RNA and the CHAMP1 gene. So knowing that, we grow the cells and then extract the proteins to check if besides the CHAMP1 gene expression, the cells also have the CHAMP1 protein expression. And then we confirm this using Western blot. So knowing that the SEH-SY5Y cell line has both CHAMP1 protein and gene expression, our next step was to do a deletion in the gene, to do a dysfunction in CHAMP1 gene, using the CRISPR-Cas9 technology. For this, we use a guide RNA with a guide sequence that, in our case, binds specifically to a region of the CHAMP1 gene. Then, when this guide RNA binds to CHAMP1, the Cas9, which is an enzyme, comes to the place and then cut the DNA, removing the complex with the scene, and then a deletion is made in the CHAMP1 gene. So for our CRISPR-Cas9 step, we used 3 guides developed by Syntego, and this was the step 1. When we did the CRISPR-Cas9 in the cell line, we obtained at first 42% of knockdown score of the CHAMP1 gene. And then our next step was to use this pool of cells with 42% of CHAMP1 dysfunction to do a cell sorting selection. In this phase, we diluted and plate 1 cell per well in a 96 well plate. So the goal was to distribute 1 cell in each of these wells to identify the colony that has the highest level of CHAMP1 knockdown and then proceed the experiments. So when we sequence each of these wells, we identified 3 colonies with 100% of CHAMP1 knockdown and then proceeded to the step 2 of the experiment. In the step 2, we treated this edited cell line using the adenovirus, the same one that we are going to use in the mice. And we did a transfection to treat the cells using the Jet Prime protocol. And we plate 50,000 cells per well and did the experimental triplicate. So we used these edited cell lines and did the transfection with the vector carrying the normal copy of the CHAMP1 gene. And after 24, 48, and 72 hours of the treatment, we collected RNA and proteins. In our first results, we can see that first, the addition we did at the cells worked, and we don't see the CHAMP1 protein expression in the triplicate for the negative control. The negative control is the edited cells treated with the reagents from the transfection procedure, but without the vector, so no vector here. And then when we treat the cells with the vector was capable to rescue the CHAMP1 protein expression in the 24, 48, and 72 hours. Also, we checked the expression of MAD2L2 and POGZ in both conditions, and we didn't see alteration in the expression, regardless the level of CHAMP1. And this was kind of expected because the literature already told us that CHAMP1 mostly interact with the protein, then regulate the levels. So our next step is to do immunoprecipitation to detect protein-protein interactions changes according to the CHAMP1 expression in this experiment. Talking about the future directions, altogether our cell work will contribute for the molecular characterization of the vector and will provide a background data for when this therapy is used in mice. Also, our previous results show vector effectiveness in restitute CHAMP1 normal expression, and with this first step, we expect similar results in mice. Cell's experiment anticipate the expected vector result after treating the mice. In the nearest future, we will have the behavior characterization for CHAMP1 mice when compared to the wild type. And also, the mice treatment with the adenovirus is scheduled to start as soon as the phenotype characterization is concluded. Our mice study is also going to infer the toxicity of the vector, which is a essential question that we need to assess for the future use in patients. With this, I would like to thank the CAG, where I'm doing my work, the Children's Hospital of Philadelphia, and all the doctors that are involved in this project helped me a lot, and also the help and the support of Dr. Håkon Hakonarsson. And also thank the CHAMP1 Research Foundation for all the support and funding. Thank you.
Just want to open up to any questions. Also to tell you there's coffee in the back if anyone needs some. You guys are troopers. I know it's a long day.
Amazing talk. Thank you very much. Actually, it's super cool. And I have like just a question about the timing, right? So how much important is the timing of these viral injections? Like, because we know that is a highly prenatal expression and then goes down, neurons tend to have like low expression, maybe OPCs, oligodendrocytes have more. Mm-hmm. So how much important is the timing of, you know, especially also for a treatment, you know, future? Thank you.
Yeah, that's an important question that we are also trying to assess using this mice model. We plan to start a treatment at around 5 weeks old, the mice, and maybe 10, and then see how it goes, the treatment. And if it's not effective or depending on the results, we can go like in a lower age or later.
How is translated to a human, basically?
Yeah, we don't know yet, but hopefully with the mice, if we can treat like, if we treat with 5 weeks and it's effective, and then with 10 weeks and it's effective, then we can do like treatment with both like child with young age, and I don't know, around 8, 10, and it worked. So it's a subjective.
Would this give us insight into what age there might be a window of treatment, and to which level it might not be? Is it safe to say it might not be as impactful, maybe? I guess we know when we get there.
We don't know yet. We are expecting with the mice to have any idea of this. So we are going to treat mice with different ages to have an idea if the vector works. So when we have this result, then we can have some idea in human.
Now it's on. Okay. So I'm just trying to think of a more convenient pharmacodynamic marker for the gene therapy. So you're trying to correct the anxiety and the behavioral defect in the mice. If you had a blood-based biomarker, in other words, does the AAV vector express the full-length CHAMP1 protein in blood cells, and does it correct some phenotype in the blood cells that would be a convenient thing to look at? I mean, you know, and don't have an answer for that, but Kozo presented today that lymphocytes from a CHAMP1 patient have radiation sensitivity, some kind of drug sensitivity. Can you correct that? And is that a more convenient biomarker than? But maybe the behavioral assay is robust and strong, and that I don't know.
Yes,
not having done it.
it's idea too. That's why we are doing the molecular characterization together with the behavior tests.
Yeah.
We are doing necropsy of CHAMP1 mice, collecting all the tissues,
Right.
and extracting proteins to compare to. So we want to compare these two levels, not only the behavior tests,
Right.
because we don't know like how different the groups are, but also see the molecular parts. So the blood cells might be interesting to look to.
Okay.
Yeah, great talk. I like to ask about the expression level. In your cell model, how the expression level of CHAMP1 compared to the endogenous level?
The expression of CHAMP1 compared to endogenous level? Yeah, the endogenous level of these cells, it was a little bit lower than when you treat with the vector. So, but there was no significant difference between them.
Okay, so in your gene therapy, can you regulate the expression level to the proper level?
Yeah, we can do that. We can try different dose of the vector.
That's right. Thank you.
Also, I won't speak to Dr. Bachbaum and his study, but they're collecting blood biomarkers, which hopefully we'll be able to answer some questions as well.
To the question around IV, so for your program, it might be worth considering 2 routes of administration. So currently, AAV9 is not considered blood-brain barrier penetrant past age 2 in humans. That's why Zolgensma is IV for under 2. So I think, to Jeff's point, mice models for neurodevelopmental disorders are incredibly limiting in what they tell us about the human condition. I think they're a good tool, more of like a bioanalytical tool to tell you whether you can express and recover some function. But take it with a grain of salt. I think humans are really resilient and evolutionarily different from mice. So I think it's a good window, but it's not going to tell you everything about once you actually move into the human condition. So I wouldn't limit that interpretation from the beginning. Use a tool for what it's positioned for, but you may, if you are considering treating very young, you may consider an IV form of administration, and then later different kind of lumbar delivery.
Yes, we are considering IV at first, and then depending on the age of the mice, intraorbital injection too. So.
Did anyone else have any questions? Okay, thank you so much. So we have one more virtual presentation, and it should